Hello people, here is a short presentation on greedy algorithm with respect to genome rearrangement. Today we will be talking about two things. One is the greedy algorithm and second is genome rearrangement. Now basically the genome rearrangement means the mutation that changes the gene content of genome or the arrangement of the gene in the genome. But how does it matter that even the position of a gene is changed in the genome? So let us take a word from our daily life such as rose. Now if you change the position of these words, the meaning of the word also changes. Similarly, if we change the position of the gene, the effect and the functioning of the genes also changes. So can we apply bioinformatics to it? Before talking about the bioinformatics aspect, let us first talk about the biology. So the genome rearrangement are the mutation that change the gene content of a genome or the arrangement of the gene on a genome. The genome rearrangement is a different class of mutation affecting a very large stretch of DNA sequence. Chromosome breaks at 2 or more locations which are called breakpoints, and this results in a DNA sequence that is essentially with the same features as the original sequence, except the order of these features have been modified. It is worth noticing that compared to the local mutation, the genome rearrangement are extremely rare. However, over time they accumulate and the order of the genes become scrambled. The closely related species usually have the similar gene order. Genes are stretches of DNA sequence that are responsible for encoding protein. Each gene has its own orientation, depending on which it is supposed to be read. Thus, a chromosome can be said as an extract, as an ordered set of oriented genes. Now, if I change the orientation of these genes, the meaning and the functionality of a particular stretch of DNA also changes. Now, if genome rearrangement and their breakpoints. If the breakpoints occurs in a non-functional part of the sequence, then the effect can be a silent effect. But if the changes take place in the function sequences, then the changes can be lethal. That's why most of these changes do not occur in a functional part, that is genes. Now, genome rearrangements have different type of models like inversion or block interchange, fusion or translocation. In the inversion, we can see that the direction and the position of the particular gene segment is inversed, and in the transposition, the position of the gene is changed with respect to other genes. In the translocation, the position of the genes among the chromosome is changed, and in the fusion, two set of genes are combined. Now let us talk about the bioinformatics problem. A greedy algorithm is an algorithm that follows a problem-solving heuristic of making a locally optimal choice at each stage with an intent to find a global optimum. That is, it locally tries to find a solution for a choice in the subproblem and then try to provide a global optimum. We can make whatever choices seems best at the moment and then solve the subproblem that arise later. The choice made by a greedy algorithm may depend on the choices made so far, but not on the future choices or the solutions to the subproblem. These depends on the permutations and the combinations. In general, the greedy algorithm have 5 components. First, the candidate set from which the solution is created. A selection function which chooses the best candidate to be added to the solution. Third, a feasibility function that is used to determine if a candidate can be used to contribute to a solution. Fourth, an objective function which assigns a value to a solution or a partial solution, and the solution function which will indicate what we have discovered in a complete solution. Now let us take an example. Here we are trying to find out the combination of number to get highest sum among the group. If we start from a number 3 and we try to make a greedy choice, the largest number in the second series is 7. So if we move from 3 to 7, the next choices that we have available are 9 and 11. Clearly the 11 is a bigger number and is the best greedy choice. However, if we want to find a globally optimal solution, we can also go from 3 to 4 to 20 and add them. Now as we know, 3 plus 7 plus 11 is 21. On the other hand, 4 plus 3 plus 20 is 27. Here we can see that the greedy choices may not be a globally optimal value. Now let us talk about the cons or the problem with the greedy algorithm. Very often the dataset may lack a global optimum value or a suboptimal solution, which may give us an hypothetical result. Also, the greedy selection may not resemble a biological selection. At times, the greedy algorithm is being confused with the dynamic programming, but one of the major changes between them is that greedy algorithms do not reconsider the solution once a decision is taken. The further subproblems are solved. Now let us talk about the greedy algorithm in genome rearrangement. In 1973, the Susumu Ohno said that the rearrangement occurs at the random position, that is, there is no fragile region. As we know that these rearrangement occurs very rarely, so they must not happen in a coding region or a region which is of some functionality. That is, there has to be no fragile region. Back then, we didn't have any proof that can tell us that these rearrangements are only happening in a particular part of a genome. But later in 1984, the first statistical argument in the favor of random breakage model was given by Naidu and Taylor. So, a test on simulated data was performed to figure out that can RDM give us some prediction power about how can we relate the number of these blocks that can be generated with the size of these blocks or the genes that can be generated. And the prediction gave us an approximate understanding of each size with respect to its number. Now, when it was compared with the real human mouse centenary blocks, it was found that the model was roughly correct and also given significant good values in real life scenarios in human mouse centenary blocks case study or cabbage to turnip case studies in the organisms which were closely related, in which the natural genome rearrangement could have happened. And because this method was dependent on the greedy algorithm, the greedy choices were made to perform the shortest way of genome rearrangement, which symbolically resembles to the natural selection procedure in the genome rearrangement or the gene selection. Now let us talk about the greedy algorithm in the sorting reversal. The goal is that given two permutations, find the shortest series of reversal that can transform one into another. Here we are talking transformation of a genome element from one to another. Now input. The inputs are permutation, that is pi and sigma, and the output are the reversal p1 to pt, transforming pi into sigma such that t is minimum. Now let us try to apply that using a greedy sorting reversal. Here is a stretch of DNA with a continuity of gene from 1 to 10, but due to some reason there is a breakage and there is a change in the arrangement of the order of the genes. Ideally, the number 8 gene was never bound to be closed with the number 3 gene, but in the new arrangement, this is so. This change in the point of reversal or the reversal introduced are called the breakpoint or the disruption in the order of the gene arrangement. Now let us see with the help of a number how this reversal can be solved using a greedy method. As we can see in the first stretch, the positive 1 and the last number positive 4, and in between stretch minus 7 plus 6 minus 10 minus. Now let us try to understand this with the greedy sorting by reversals. As we can see here, we have a stretch of numbers and let us assume that each number represents a gene. In the first series, we can see the positive 2. In the first case, as we can see, the positive 2 here is not at the correct position. So we shift it at its correct position. And now we can see the positive 2 has a negative sign. We change it to positive 2. In the second series, as we can see, in the third series, here we can also see the negative 3. As we can see here, negative 3 is also not in the correct position. So we shift this position and change this to positive 3. Also here, we perform similar iterations to the FS sequence until an ideal solution is created. It took 12 iterations to understand the ideal condition for the gene order in this case. Thank you.